I am presenting the results of the NILE study, which was a prospective clinical trial comparing standard of care tissue testing for patients with metastatic non-small cell lung cancer for molecular profiling with an NGS that was performed at the same time at study entry on blood specimens obtained from the same patients. This is the first prospective clinical trial to compare NGS performed via a standardized, highly sensitive assay in the blood, plasma, with standard of care tissue testing. And with standard of care tissue testing, the protocol allowed means that the patients were allowed to be profiled with whatever was the modality of choice of the treating physician or treating institution. The clinical trial's primary endpoint was to assess non-inferiority of the plasma liquid biopsy compared to the standard of care tissue testing, and the clinical trial proved indeed that the NGS testing in the blood is non-inferior to the standard of care testing. It actually performed equally well. Standard of care testing discovered the molecular alterations in 67% of the cases versus 87% of the cases with plasma NGS or liquid biopsy. And another important characteristic of the clinical trial was that the turnaround time of the plasma assay was significantly shorter than the standard of care tissue testing, 9 versus 15 days as a median. The results of the clinical trial confirm what many users of liquid biopsy in the academic community already knew, but it is important in the sense that it was designed and prospectively executed to answer this question. Another component of the clinical trial secondary endpoint is to look at the outcomes of patients that were molecularly profiled, to look whether there are any benefits for patients that were profiled with plasma NGS, because the test returned faster and we discovered more alterations. That would consolidate our impression that this assay can be offered as the first assay for patients, followed by reflex tissue testing in the scenario where no mutations are detected in the plasma NGS.